let's talk about the third class of organic compounds, which are proteins. Proteins, we should know, are chains of amino acids, so that subunit, that monomer is amino acids. And again, carbon, hydrogen, and oxygen, but now we've got a nitrogen component, which really is one of the things that makes proteins distinguishable. They have nitrogen, as well as occasionally sulfur. Proteins have a wide range of functions, and some of those wide range of functions are things like they have a structural component, right? And by structural component, we're talking about the fact that a large portion of your body is made up of protein, like muscles, as well as things down to like within the cell membrane, there are proteins embedded for a structural component. Protein can also be an energy source. We know that we eat foods with protein, and that provides our bodies with energy in addition to the carbs and lipids that we're consuming. There are proteins that are chemical messengers. Right? Those chemical messengers, we typically refer to them as hormones. Proteins are also a part of your protection, your body's protection. Antibodies in your immune system are a type of protein that are helping protect your body against foreign invaders. They help with transport within your body. For example, hemoglobin in the blood within the circulatory system. Hemoglobin is going to help to transport oxygen throughout the body. And one of our really big groups of proteins are the ones that catalyze reactions. And we should know that those are those enzymes that we so often talk about. So when we talk about proteins, like I said, they're chains of amino acids. And the interesting thing about amino acids is that a huge portion of an amino acid is pretty consistent. That being the amino group, which is where that nitrogen is found, and then the carboxyl group, which often we don't deal with the carboxylic acid, we do carboxyl group, all right? But it's actually that R side chain. And I just want to point out that R does not stand for any sort of element. R is just referring to the idea that it's a variable, right? The idea that it's something that changes. It's the distinguishing feature, right, for amino acids. So the variable determines the amino acid, meaning whether it's methionine, whether it's leucine, or any of our other amino acids, it's really all dependent on that R group. We should know that amino acids are linked together by peptide bonds, and that peptide bond occurs between the carboxyl group of one and an amino acid group of another to form a polypeptide, and that's just a fancy word for a protein. Poly, many, peptide type bonds. Many of those bonds formed together between amino acids creates the protein. So we have one amino acid here and a second amino acid here, and you can see that we have a carboxyl group versus an amino group, and the bond is taking place between those two groups, creating this water molecule. And what you end up having is a connection, that peptide bond from the carboxyl group to the amino group. One of the most confusing things about proteins is their conformations, right? Talking about their figures or structures, how they're arranged. We're talking about the 3D shape of a protein. We've got a primary structure, which is really just a chain of amino acids. And that chain determines the protein function. If amino acids are in different orders, the protein that results is going to be a different protein. But then that chain is going to coil or fold on itself to create a secondary structure. That coiled or folded structure is then going to continually fold upon itself to create a tertiary structure. And it becomes to have this 3D knotted shape. Right? So we go from having our primary structure being just our linked amino acids to our secondary structure may start to have a coiled appearance. That tertiary structure, that coiled appearance is going to kind of start to not upon itself. And then some proteins are going to have a quaternary structure, and that quaternary structure means that a protein with a tertiary structure bonds to a protein with a tertiary structure, bonds with a protein with a tertiary structure, and those multiple proteins bonded together, so multiple polypeptide chains, end up connecting together to give you kind of like this massive protein structure. This picture just depicts our primary level, secondary level, tertiary level, and then quaternary level. You can see here that we have one tertiary structure bonded with others. Our last group of organic compounds are nucleic acids. And we should know that the monomer for nucleic acids are nucleotides, composed of a sugar, phosphate, and a base. Again, sticking with the carbon, hydrogen, oxygen, nucleic acids have nitrogen, which is going to be that base portion, and phosphorus, which is going to be composed in that phosphate component portion. These are going to carry the instructions for cellular activity, as well as a wide variety of other things. This picture here should be familiar, representing our sugar, phosphate, and base of a nucleotide. So, we can compare two of our nucleic acids, DNA and RNA, being deoxyribonucleic acid, and if we look, they should be relatively the same except for deoxy without oxygen. There should be one little difference there. DNA is missing one oxygen molecule. DNA is a double-stranded helix, while RNA is just a single strand. DNA stores that genetic information, while RNA is what we use for protein synthesis. Remember, we take that genetic information and we convert it into RNA to create those proteins. Our sugar in DNA is deoxyribose, again, our ending being ose, while RNA is just ribose. And our nitrogenous bases are relatively the same, with the exception of thymine being replaced with uracil in RNA. And we should know that A goes with T, unless A is going with U, and G goes with C. So picture, right, we have DNA showing how we bond together between our bases versus RNA being that single strand. Another one of our nucleic acids that we often forget is ATP, adenosine triphosphate. Adenosine triphosphate isn't a chain, it's actually one single nucleotide. So it's only one nucleotide large or small. And this is our cellular energy. Our sugar is going to be ribose. Our base is always going to be adenine. So instead, we actually don't have thymine, guanine, cytosine, or uracil in ATP because adenosine triphosphate is named after its base, adenine. And then the interesting thing is that in ATP, we have 3 phosphates, and this bond between the last phosphates is where that energy is really stored. This should kind of be a review from a lot of the information that we talked about in biology. This pretty much wraps it up, guys, for our lesson on organic compounds. If you have any questions, please feel free to see me.